Welcome to today's webinar, The Cell. This is where everything happens. This is one in a series that we're going to be producing, and I produced some in the last year since I joined Taurus. And this one is sort of one of the basic things I talk about with farmers, and a lot of people wonder why I talk to farmers about cells. Well, I think I'm a believer that, you know, the more we understand about plants, the better we will be at growing them and fertilizing them. So the cell is an important part of it, and everything happens in the cell, and that's what we're going to focus on today. Before we get into that, I kind of like to show this photograph because we have this canola field, and this is Phil Thomas standing in it from years ago when he was a much younger man. And we know that these plants up here are dealing with different things in the rooting zone than plants down here, both in water availability, nutrient distribution, pH, organic matter, all kinds of variability, and yet they have all the same genetics. So plants up here are regulating themselves, and plants down here are regulating themselves, and up and down regulating genes to deal with what they're facing both above ground and below ground. And they're all trying to get to a state of homeostasis or balance in the cells which make up the plant, and that's where the cell comes into play. And it's about cells. Here's a typical cell. There's a lot of things going on, and we're going to talk about all these little organelles inside the plant and the cell wall and plasmodesmata. But really what it comes down to is he who builds the most cells wins. Because the way we are producing crops here in Western Canada, outside of forages, we're producing annual crops. So all of our crops are programmed to die genetically. So as we grow this crop, from during the vegetative stage up until reproductive stage, and then we have it fertilized and we start filling those seeds. It's this part of the curve, this growth phase, which loads the plant with the nutrients in the leaves and stems and roots, which will then start being moved into the seeds, because it's the plant's objective to produce good, healthy seeds, and that's what we want them to produce and sell. So it's all about this growth in cells that is important. So today we're going to talk about cell types and how they grow and the functions of them. We're going to talk about the cell wall structure, or cell structure, including walls, organelles, and sort of the guts of the cells, and the membranes, because that's what regulates what comes into the cell and goes out of the cell in all parts of the cells, and that's where we'll end it. And it's these cells that are important in terms of regulating the nutrient flow and water and so on. So let's start at the beginning, the cell types. Well, where do cells get born, so to speak, putting it in the human sense? Well, cells get born here in what's called the apical meristem, the growing point, and this is just canola. So these are the cotyledons up here, and this is just a split. There's two cotyledons, and right at the junction of that is the growing point. And most of us know that if we get a frost in late spring and we kill this growing point, which turns black, the plant dies, because this growing point will differentiate into the future leaves, the stems, the seeds, and so on. And this is canola. This is just showing you soybeans, and this is the growing point in soybeans. Again, you can see the vascular system here that's feeding nutrients and moving hormones around, and a lot of hormone production takes place in these growing points to communicate throughout the plant and with the root. Here's the growing point in the pea plant here. And all these cells eventually wind up making tissues. And there are three kind of basic tissues in plants. There's dermal tissue, the stuff around the cells and around the leaves. There's ground tissue, the tissue inside the plant. And then there's the vascular tissue, or those tubes that make up the xylem and the phloem and move nutrients up and down in the plant. And you can see here's the root, and then we have a sort of a different kind of structure, because now we got these tubes that are running up and down the stem. And then when we get into the leaf, you can see that the veins, which is the vascular tissue, run into the veins, and we have the dermal tissue here, or the ground tissue here, sorry, and the epidermal or dermal tissue. And I'm going to show you exactly what this looks like with my own photographs in a sec. So this is just showing you a dicot, which are now called eudicots, like a soybean, which I'll show you. And here's some showing what happens in monocots or grasses. And lastly, this is sort of a cross section through the stem, showing you xylem and phloem here, and fibers and all these kinds of tissues that are in the stem. And now let's take a look at them. So here we have a wheat root, and you can see that this is the xylem and phloem here, and you can see the actual tubes that run all the way from the root tip all the way to the tip of the leaf. And then there's tissue around it, and then the root hairs are out here pulling in all the nutrients and exuding organic chemicals, compounds into that rhizosphere to feed microbes in there. So here we see a cross section through a soybean, this one and this one. This is a little closer. Let's look here. You can see the dermis here. This is the ground tissue, and then this is the vascular system right in here. Here's wheat. You can see the same thing. Here's the dermal tissue. Here's the vascular system, and then this is all that ground material. So it's very common. And when we take a look at the leaf, I want to point this out so I can better explain things like photosynthesis later. You can see that on both sides of the leaf, we have sort of a dermis and a cuticle that's waxy, and we have up here the epidermis and the cuticle, and that waxy material that covers it to prevent the loss of water through transpiration. In that tissue, we have formation of leaf hairs and trichomes and those kinds of things. The photosynthesis takes place in these chloroplasts, these cells right here underneath the epidermis, which can gather sunlight even at that level. So you want those in the palisade mesophyll and then the spongy mesophyll down here. Now, the plant moves its water and gases into the spongy mesophyll, and you can see there's a lot of air space around it. So it can diffuse gases and water vapor into that space, and it can kick out that water vapor that drives the whole transpiration system from the root tip up. Water leaving the leaf puts suction on the root tips and the root hairs, and it pulls it up and it leaves through those stomata. You can also see that right here there are guard cells. That's our first cell. We see that right around the stomata there are specific cells called guard cells. And there are about 40 different kinds of cells in plants, from chloroplasts to guard cells, to spongy mesophyll, palisade cells, and so on. And then we have the vascular system right here with the bundle sheath around it. We have the xylem here above and the phloem down below. So that's just gives you an outline of the leaf when I talk about it. This is a cross section through the leaf, and this is, again, a look at the vascular system, and here's what it actually looks like. You can see this is the soybean leaf, and I've cut through the leaf just up from the stem of the leaf, and this is the main vein, and then there are smaller veins that run up there. And as we magnify that, you can see that we have the vascular system, the dermal system around here, and then the ground system. So it just moves everywhere. And then when you look really close, you can see the phloem and the xylem right here in that plant, and it's just connected through the stem, as we've seen, all the way to the roots. And I just showed you this. These are stomata on the bottom of a wheat leaf, and you can see a pile of them, and you can see it's a tiny hole there. And if you look at them, here are the guard cells right here. This is maybe a better one, the guard cells here. And then there's the opening, the actual stomata right there in the leaf. And it's these guard cells that are important. They store things like potassium and calcium that are involved in opening and closing the stomata in there. And then via transporters, they can close, stores are used to open and close those stomata very, very quickly. If we take a look at a root, here's a diagram of a root, you can see that the root hair is an individual cell. It's one cell. So we have root hair cells. They're individual cells, and they only live about 5-6 days. But if you kill a few of them off, it doesn't hurt them. But you take a look at diseases like clubroot and aphanomyces, they actually infect the root hair, and that's where they get a hold on it. I will point out one other thing is, if you notice, the root hair takes in nutrients with water, and it moves it through these little portals between the cells called plasmodesmata. It can also move nutrients in between cells, which is in between cells, but when it hits this barrier here called the Casparian strip, which is impermeable, the nutrients and water have to move into the cell and then pass into the xylem. Or it can go directly from cell to cell to cell this way, and I'll show you those a bit later. But the root hair is a single cell. So you can see this germinating wheat plant, that each one of these is an individual cell. And I will talk in another webinar about how the root hairs and the pollen tubes actually grow. Here's another kind of cell nobody much talks about called a pulvinous cell. And these reside right at the node of plants, whether it be wheat plant or canola plants. And you can see that they look like muscles. So when a plant falls down due to lodging, the pulvinous cells have the ability to differentially expand. So when you notice a wheat plant falling down and it suddenly tips up, it'll tip up at a node. And it's because these pulvinous cells expand and they're pressurized largely by potassium and water, and they push the plant up. So cells perform a lot of different functions. And here we can see that this is a cross-section through the stem of a corn plant at about the 2-leaf stage. And you can see we already have the anthers formed here. We have the xylem and phloem all in here. This is the cob that's forming here. And we have all of these leaves, which all these cells, you can already see the vascular system is formed in all of these leaves. So the plant by this stage has all of its leaves formed and they're curled up around there, and then they just have to unfurl. So it's all of these cells that make up the plants, and they're different cells and they did different nutrients because they have different functions. So to get homeostasis or balance in a plant is no easy topic. And that's what we try to do with nutrients and soil testing and plant tissue testing, so that we can see what the plant is telling us. So how do actual cells grow? Well, they're born up in that apical meristem, or in the root tip, in something called the quiescent zone right near the tip of the root. So there's 2 types of cells. There's tip growth cells, and there are only 2. And one of them I've already talked about is the root hair, and the other one that is a single cell is the pollen tube. And they grow by sort of streaming the cytoplasm, and I'll be covering exactly how that works later or in another webinar. So that's one. And then there's diffusive growth, diffusion. So most of our plants go vertically, upright. So you can see that you want the plant to differentially grow in a vertical position, so the cell wall of those cells are such that they will containerize them through a bunch of fibers, so that when the cell is expanding, because the cell is extremely small, it actually grows longitudinally more than a cross section. And I just show you this is another root here, but here you can see cells in the wall of the stem demonstrating that kind of diffusive growth, and they grow at different rates. You can see them all through here growing that way. In fact, here you can see this is in the tip of a radicle that's just expanding all these tiny cells, and they will just be expanded just like a root going through the soil. Be no different. The root going through the soil expands these cells, and by hydraulic pressure actually moves the root tip through the ground. And in the end, for us, it all comes to nutrients. And I just throw this in. I think I got this from Tim Myrick down in the States. But it just shows you that within the cell, for example, the mitochondria, we have iron, we have phosphorus. Here we have manganese, we got ATP being formed here. We got all kinds of DNA going on. In the chloroplasts, we have a lot of copper, a lot of calcium, a lot of magnesium, a lot of iron, and I'm going to talk about those specifically. So every one of these little organelles in here have different requirements. For example, up here in enzymes, we have molybdenum, copper, zinc, those transition metals that are used for electron transport and for oxidation reduction processes. And then we have the nucleus with the blueprint in there for all the all the genes. And we have zinc, which is used to open up the DNA so that the messenger RNA can make copies of it. So all these nutrients combined make the plant work, and it's cell by cell. And the important one here is the vacuole. The vacuole is I'll cover it now. I'm going to cover it again in a little while. But the vacuole is equated to a fat cell. As a human, you can't get rid of your fat cells. So when you eat too much and you don't use it up, the fat cell gets bigger. And if you're starving, it shrinks. Well, the vacuole is exactly that. And this isn't in proportion because the vacuole will actually take up about 80% of the actual cell space. But the vacuole is where the plant stores nutrients that are in excess or toxic materials that it wants to prevent from damaging it. And we're going to talk about those in a sec. So what happens in the cells? Well, these are sort of similar things, but quickly, let's look at sulfate. We'll use sulfate here. Sulfate is taken up through transporters, proteins in the root. This is the root cell, and we take that sulfate up and we store some in the vacuole in the root, and we showed you that in the root. The rest of it goes in here. Some of it is used, and it makes cysteine, which is the sulfur-containing amino acid, and then methionine is made from cysteine. The rest of it moves up through the xylem, again through transporters into the leaf. In the leaf, some of that will move into the vacuole for storage of it. Some of it will move into the phloem and be recycled back through the whole system. The rest of it goes up here as sulfate, and it's reduced because the plant can't use sulfate. It has to reduce it to sulfite, then sulfide, and then it forms cysteine, and from cysteine it forms methionine, but also forms glutathione. Glutathione is really key in stress management in plants, and in my stress presentation, you'll find out all about glutathione. And glutathione is then circulated through the plant, and that's what the plant monitors for its level of sulfur in the plant. And then some of that goes into the mitochondria where it's used as well. And the same thing happens with nitrate. It's taken in as an NO3, and it's got to be changed right here. We go to NO2, and then nitrate reductase, which contains molybdenum as a cofactor, and one of the reasons why we have just recently for 2020 included molybdenum in our build product, and then into ammonium, and then it's incorporated in the chloroplast. That's where all this takes place in the leaves, some with sulfur in the roots. So that's what happens in that cell, in the chloroplast cell. So let's now take a look at the walls, the organelles, or the guts of the cell quickly. So here's a typical cell. We have a nucleus here, which is sort of the brains of the whole system. Here's that vacuole, that storage system. Here are the chloroplasts where we have photosynthesis. Here are the mitochondria where energy is produced. Here's something called the Golgi apparatus, which are involved in packaging up things and moving them around. And then there's the endoplasmic reticulum right here, which is sort of a channel and a system that runs throughout the cell. So that's all there is. You know, and I learned this, I remember back at university in 1966. I never understood it then. It took me till now to understand all this. And then one other thing I want to point out here is this middle lamella. That's right there between the two cells. And this little blue line is the plasma membrane, which sits inside of the cell wall. So let's take a look at the cell wall. Well, the cell wall has many functions. It gives it structure. It's rigid. It contains lignin and all kinds of hemicellulose, cellulose, and microfibrils. So it gives it structure. It protects the cell against invasion. It regulates the flow of things in and out of it. But let's take a look at the important part of this. There's a middle lamella here, which is the part right here. I'll show you on this one, it's better. See, there's a middle lamella here, and then there's one cell here and one cell here. And this is just like a gelatin, so it slides, because when cells are growing, they're growing at different rates, and they got to slide by each other as they expand, and that's where it happens. And calcium is really key in that. And these little dots are all calcium. So we have this heavy load of calcium right here in the middle of the lamella, and a lot of it in the vacuole. Calcium is highly regulated in plant cells because it's used for not just cell wall structures. Its primary use is a signaling material or element, and it regulates it so that if it's attacked or there's a problem, it can flood it into the cytoplasm and use it to signal the cell that there's something going on and respond to it. So back over here into this. So we have layers between the plasma membrane, which is the membrane on the cell, and the outer membrane here, or the middle lamella, and then we got the primary cell wall here. Now, these bars here are cellulose microfibers, and I want to point out this here, this thing called rhamnogalacturon is a sugar which contains most of the boron in the plant. So the combination of calcium and boron is what gives you really good cell wall structure. One without the other cannot work very effectively. And these microfibrils in here are like rods in cement, rebar in cement, and then the rhamnogalacturon, which I can never pronounce properly, is actually very linked to holding these microfibrils together. So that's the importance of the cell wall. And here's just a shot showing the vacuole and the nucleus. Here's that middle lamella. You can see that that's where two cells together, and that little line in between is the gelatin that allows them to slide back and forth past each other as they expand and contract. Or if a wheat plant is bending in the wind, those cells have to be able to move past each other in order to be pliable, or they're going to break. So that's really key to the plant. Let's look at the guts. The important part of the gut here in the plant are the chloroplast, or the plastids. And we can see here's a chloroplast, a chromoplast, which gives you color, an amyloplast, which in wheat, for example, where we have amylase is the starch, or amylopectin is the starch in wheat endosperm. They're stored in the amyloplast. So the plant has to make the amyloplast, and then the plant fills it. So that happens right after pollination and takes place. So these are all the various plastids and different kinds of things that are going on, but I'm going to focus primarily on the chloroplast. And here's, again, just to remind you, here are the chloroplasts sitting here within the mesophyll, or the cell tissue just below the upper surface of the leaf. So photosynthesis is just a function of taking CO2 and water, breaking it down, making a little bit of ATP, kicking out the oxygen, and taking some sunlight and producing sugar. It's very simple. So we get water going in here, we get sunlight coming in into photosystem 2. Photosynthesis starts in photosystem 2. We then move some of those electrons that are excited over to something called the Calvin-Benson cycle, where we take in CO2 with an enzyme called ribisco, and we produced sugar, glucose. That's all there is. But the secret is in how it transfers electrons in order to make the ATP. So here's quickly, you don't have to memorize this. I'm not going to have an exam on this darn thing because most of you can't do much about this, but I need to talk about the nutrients that are involved in this process, because without this process, the plant does not perform and do anything. So first thing, you break the water molecule, and I'll show you how, and and the hydrogen is used here to help make some ATP, but sunlight activates, energizes this electron. This electron is carried with a compound called plastoquinone through the cytochrome, through the membrane, and passes it on to this thing called plastocyanin. The plastocyanin then carries it on through to photosystem 1, and it's transferred by ferredoxin. Ferredoxin is cofactored iron, plastocyanin, copper. About 50% of the copper in a plant is used specifically for the electron transport here. We all need iron in both here, in photosynthesis and in the mitochondria for production of ATP and energy. And those are the key reasons we have those here. They're involved in a lot of photosynthesis. And all we get from that is a little bit of ATP. So here's some of the key things. The chloroplast, central atom, magnesium, surrounded by nitrogen. That's why we need a lot of nitrogen when we're building plants. We need magnesium for this process. We also need magnesium to activate ATP. And for those of you that maybe have an economics degree and never learned what ATP is, adenosine triphosphate, that's the energy package used in all life. And most of it is actually referred to as magnesium ATP. So magnesium is key to activating ATP, and ATP contains these three phosphorus, so ATP and phosphorus go work hand in hand. Well, how did the plant break down that water molecule? Right here. It's a process called the Hill reaction, and here are the actives, manganese and calcium and oxygen. But manganese is key. So if we have low levels of manganese in the plant, the process of photosynthesis goes in the toilet and we don't get much of it done. Here's ferredoxin. You can see ferredoxin has iron, sulfurs, and more sulfur, but here we have cysteine, amino acids. Here is plastocyanin. Copper as the main active ingredient in this case, a bunch more nitrogen and a bunch of sulfur, including methionine and cystenate. So reason I put that into perspective for you is that photosynthesis is just not magic. It's magic if it's got the right nutrients, and it's magic if the plant isn't stressed. If the plant is stressed, then actually it produces a lot of free radicals and it becomes toxic to itself. And you'll learn all about that when we talk about stress management in one of my existing webinars. We're going to take a look at what's inside that plant. And here we go again, the vacuole. We have mitochondria for respiration. I've covered a little bit of that. We have Golgi, which package up things. Peroxisomes, we'll talk about it another day. We have the middle lamella, and now we're going to move on to this little blue line here called the plasma membrane, and we've got chloroplasts. And here's, I just throw this in for some of the folks that like to get a little excited about how a plant actually up and down regulates genes. Well, what happens is that if there's a signal that's sent and the plant decides it needs to do something, it can't run away and hide, and it can't move to a place where there's more water or less water, or where there's more nutrients or it's not compacted. So it has to do something. So what it does, it goes to its blueprint here in the nucleus. And something called messenger RNA makes a copy of it. And we'll use phosphorus here because phosphorus is pretty key, and plants will do everything they possibly can to get phosphorus. So there are phosphate transporters here, which are proteins, and some of those proteins are actually enzymes which can break down phosphorus in the soil in either organic matter or as elemental phosphorus. Makes a copy and it runs it down here into something called ribosomes, which are stuck onto something called the endoplasmic reticulum, which is this river that runs through here. And the ribosomes make copies of that gene, hundreds and thousands and millions of them, very, very quickly, runs them into the endoplasmic reticulum and sticks them into the plasma membrane here if it's root cells, and the root cells are all doing this, and they're pulling nutrients from the vacuole, and they're all connected to the mitochondria, which is providing energy, sticks them in there, and it excretes either enzymes or molecules and acids into the rooting zone to extract nutrients, and then pulls them in to feed the cell. Now, here's something I want to point out. You take a look at this endoplasmic reticulum and look what happens. It actually moves between cells through those plasma desmata I showed you in the root hairs. or in the roots, in the lateral roots and the root hairs, and moving into the xylem. So the other thing that can happen here from a genetic point of view is plants now we know make something called microRNA, which are small parts of RNA, and they can move it through this endoplasmic reticulum into cells adjoining it, and it spreads like a fire through the cells, and we get a systemic reaction to, say, an invasion by a disease organism. So the plant can communicate using the microRNA and hormones and respond to whatever environment either is affecting it, either abiotic or biotic, or living organism kind of infections. Lastly, the membranes. This is that plasma membrane that sits inside the cell wall, and it regulates what goes through it. First of all, it's almost impermeable. Some gases can go through, alcohols can go through, water can go through slightly, and urea can go through, and that's why people put urea, for example, in some herbicides, or we can use dissolved urea as a foliar nutrient and get penetration because it has no charge. So this small or uncharged molecule can go through the plasma membrane of the cells and through the cuticles and pores in the cuticles and through stomata and get into the plant. But look at this. Fructose is impermeable. All these ions can't move through. None of these charged molecules can move through. So this is a barrier, and all these little blue dots are phosphorus, and these are lipids in here. So it's hygroscopic, or it prevents the movement of things through this membrane. So what the plant does when it makes those proteins based on the plant sitting in the nucleus, it takes them and shoves them into the plasma membrane. So some go right through and can allow portals and transporters that can allow nutrients and water through, and they have sensors in there so they can measure everything that is going on. It's amazing to me. And it even has different kinds of transporters when it's hard to get a nutrient, and here we're using potassium as an example, they have something called HATS, high affinity transport systems. When there's lots of it there, say a root hits a sideband or it hits a granule, well, there's a heavy concentration there, and it doesn't have to use much energy, so it uses low affinity transport systems. So it can even regulate that. Even water is controlled through proteins called aquaporins. There are about 30 different kinds of aquaporins, and you can see this is that plasma membrane again in the cell. So even water is controlled, both going in and out, and if there's too much, stored in the vacuole. So here, let's take a look lastly at what happens in the cell in terms of nutrients. And this is, this brown line here should have been probably blue in this book, but this is the plasma membrane, and this is the vacuole. You can see a couple things. Number one is the pH, about 7.2, and in the vacuole, it's acidic. So when you have a difference in pH, you have a difference in pressure or potential to move through electrogradient across here. So things can move back and forth, but they have all these transporters stuck in these cell walls or cell plasma membranes, the tonoplast, and they have all of these transporters stuck into the plasma membrane. So let's take a close look at this. So for example, you don't have to worry about these names, they're antiporters and so on. So you can see that there's a transporter here in the vacuole that's for calcium, for cadmium, for magnesium, for sugars, for nitrate over here. Okay, and at this side, there are gated channels that move nutrients, anions, cations, and so on, in large volumes, potassium, calcium, and so on. So as I said before, the vacuole storage, when the cytoplasm is out, it'll move stuff in there. When there's too much, it'll store it in the vacuole, and the vacuole can expand to and just push all those other organelles up against the cell wall. So we have this phosphorus, nitrate, just a repeat of this, and we have all these moving in and out. Now, the last thing I'll say here is in channels. For example, they're called gated channels somewhere, they have various names. But where we have channels, channels are a little different. Channels are used where they want to move massive amounts very quickly, and the best one is in the guard cells of the stomata. When a plant is short of water, the root tip sends a signal using a hormone called abscisic acid up into the tissue, that hormone, and that tissue will generally cause a closure of the stomata. The plant wants to have this happen fast, so those guard cells are full of stuff, and when they want to close them, they use these gated channels to move that potassium and chloride and some calcium very, very quickly in order to bring about a response. So there's different speeds at which all of these transporters can work, and different amounts of energy that the plant will expend depending on what it senses in its environment. So there's your introduction to cells, what some of them do, some of the structures that are in there. I hope you learned something. I hope you enjoyed it. Some people think this is a waste of time and it's too complicated. Well, when we get into really understanding plants, and I think we've really done plants a disservice by not understanding them. And I think it's, from my objective, is to try and educate people and get them excited as I am about how plants actually function. So look forward to seeing you at my next webinar. Thanks a lot.